Yes, so I would like to introduce our next presenter who will be presenting on systems and synthetic biology to build programmable cells and a sustainable bioeconomy. The presenter will be Tae Seok Moon. I think I'll just say Mr. Moon to make sure I'm not getting any other pronunciations wrong from Washington University. So, Mr. Moon, Professor Moon has 24 years of research experience in chemistry, metabolic engineering, systems biology, and synthetic biology, including 5.5 years of industry experience. During his career in the biotech industry, his chemical derivatization technology led to the synthesis of novel biopolymers and for patent applications. As a multi-team project manager, he coordinated preclinical studies and helped launch four products, including biopolymers for osteoarthritis treatment, ophthalmic surgery, injectable fillers for wrinkle correction, and biodegradable materials for drug delivery. For commercialization, he also participated in bioprocess scale-up, construction and operation of commercial-scale bioreactors and downstream facilities, and submission of a drug master file to the US FDA. During his academic training at MIT and UCSF, he has built expertise in metabolic engineering and synthetic biology. His research focus that from 2012 to present with about 15 external grants, $7.3 million of external funding to Professor Moon, $16 million to the entire teams. His research focus basically is understanding gene regulation, evolution, and metabolism, and building RNA regulators, biosensors, and genetic circuits. His current projects include engineering probiotics for controlling neurotransmitters or modulating the gut or skin microbiome, investigating changes in the regulatory network of engineered rhodococcus through multiomics for waste valorization, establishing a generalizable model for predictable RNA regulator design, and building complex RNA-based genetic circuits, developing kill switches for biocontainment of genetically engineered microbes. He has published 52 peer-reviewed papers, has filed 9 patents, has given 49 invited and 118 contributed presentations. He has advised 26 permanent PhDs, stroke postdoctoral, and 28 undergraduate researchers. He is a founder and head of the Scientific Advisory Board of Moonshot Bio. Several awards include a B&B Daniel I.C. Wang Award, an NSF Career Award, an ONR Young Investigator Award, a John C. Sulada Fellowship, an ILJU Foundation Award, an LG Chemical Fellowship, and the SNU President Prize. He currently serves as an MIT Educational Counselor, an external evaluator of Seoul National University, an editorial board member of multiple journals, a conference organizing committee member of SEED22 and ME15, and the council member of Engineering Biology Research Consortium. He is the founding chair and organizer of Synthetic Biology Young Speaker Series, a weekly multi-year seminar series in which a global thought leader, a synthetic biology pioneer, and a young rising star discuss synthetic biology, systems biology, and metabolic engineering with more than 1,000 global audiences. Well, that's the rich profile of Professor Moon. So at this particular point in time, I would like to invite Professor Moon to deliver his keynote address. Professor Moon.
Thank you so much, for very nice and introduction. Can you hear me okay?
Oh yes, we can hear you.
And then can you see my slide, as a presentation mode?
Yes, we can see it.
Okay, thank you so much. So thank you so much, and hello everybody. My name is again Tae Seok Moon from Washington University in St. Louis. I really thank the organizer for this talk invitation. Dr. Hesser, already gave, his wonderful overview regarding synthetic biology, and I have also observed as a synthetic biologist the beginning of synthetic biology in year 2000, and its growth since then for 21 years. I'm seeing the exciting start of synthetic biology in Africa and I'm honored to be the part of its start. Today, I'd like to talk about our efforts to develop a novel microbial host for upcycling waste polyethylene terephthalate. That's one of the plastic waste. And then I'll share some unpublished data. I envision our future as follows. We engineer microbes that take waste as inputs and generate value-added products as outputs. Here, waste can be plastic waste or even some plants, and also even CO2 in the atmosphere. And then, of course, product would be biochemical and some biopolymer, also high-value chemical or pharmaceuticals. One of the critical waste is the plastic, and our goal is not such beautiful PET recycling, but PET upcycling. Our process is a hybrid process consisting of a hydrolysis step to generate TPA and EG monomers and microbial conversion of these into value-added products. We are interested in waste PET upcycling because waste PET represents 8% of global solid waste, and the current PET chemical recycling suffers from the relatively high cost of hydrolysis, monomer purification, and its repolymerization compared to the virgin PET price. In contrast, our process uses a novel microbe that is flexible to potentially produce any desirable products, allowing us to cope with market price fluctuations. Today, I will demonstrate two higher value products as a proof of concept. There are two main challenges, of course. For shorter hours, and then use our microbe to produce value-added chemical without any purification step except just dilution. Our preliminary economic analysis to obtain 90% monomer yield showed both technology would similarly cost. The second challenge is efficient monomer bioconversion. The main biological issue is that only a handful of reports demonstrated microbes capable of growing on both TPA and EG. In addition, 2 more NaOH per 1 mole released TPA is required to maintain pH 8, so the resultant media would have high osmolarity, limiting high TPA and EG loading. The state-of-art technology demonstrated PET to PHA biopolymer conversion using purified LCC to degrade PET into TPA and EG at 70 Celsius for 24 hours, and then an evolved Pseudomonas strain to produce PHA biopolymer. In contrast, our technology required neither purification nor sterilization of the NaOH hydrolysis products. Importantly, our strain used 1 order of magnitude higher monomer concentration despite high salt levels due to used NaOH and HCl for utilization. Let me introduce our novel high salt tolerant Rhodococcus strain named rPET for the first time. We identify it by screening many wild-type and mutant bacteria, including Pseudomonas and Rhodococcus, and show up to 37% biomass yield per used PET plastic, utilizing both monomers as sole carbon source. We have been developing many genetic tools for a related species Rhodococcus vacus for 9 years. Fortunately, these tools are functional with some optimization in rPET. We first asked why rPET can use high TPA concentration. To answer this, we perform comparative genomics and functional analysis. Unlike other TPA utilizing strains that are assumed to have only one TPA catabolic pathway, our analysis suggests 3 redundant TPA pathways via 1, plus 2, or 3 pathway, explaining its robust growth at high TPA level. For example, knockout mutant M1 showed growth on TPA despite a long lag phase, implying the presence of unknown PCA decarboxylase. Interestingly, we observed PCA accumulation up to 2 days in M1, and then consumption of PCA, implying PCA accumulation activates unknown PCA decarboxylase. Furthermore, when we supplemented phenol or benzoate that is degraded via the cat branch of beta-ketoadipate pathway, we found interesting biphasic growth for using phenol or benzoate and then TPA later. Based on our analysis, we built a metabolic map. To confirm this map's accuracy, we decided to grow the triple knockout strain on multiple compounds as its sole carbon source. For example, as expected, no growth was observed when TPA or muconate was used, while we observed 100% muconate yield per benzoate when glucose was supplemented as the growth substrate. Given these results, including the long lag phase of the M1 mutant, we hypothesized the native PCA decarboxylase is the bottleneck step, and we are currently optimizing heterologous PCA decarboxylase for efficient TPA to MA production. Interestingly, we from the PET hydrolysis products. In summary, we discovered a novel salt-tolerant microbe that grew well up to 0.3 more TPA and 0.3 more EG. In addition, we built its metabolic map for PET utilization by comparative genomic and knockout analysis, and demonstrated that high-value keratin can be produced from the PET hydrolysis product. In parallel, we are performing protein simulation assisted directed evolution to engineer mesophilic and thermophilic PET degrading enzymes. This approach basically complements our NaOH hydrolysis approach. We observed PTAs containing lysate supported rPET growth and identify mutation target for improved PTA's activities both at ambient and high temperatures. I wish I could share this data with more time, but I will wait for the next opportunity or through publication. We have also built a bigger metabolic map based on omics and preliminary metabolic modeling, which will help expand our product portfolio. These are the summary of our current project, but I cannot cover all today. With that, I'd like to thank all my lab members, especially Dr. Diao, Drew, Dr. Davis, Yifeng, Rhiannon, and Jordan. Also, my two advisors, Chris and Chris, for the, of course, all the advice during my academic training 10 years ago, and all the current funding agency, especially DOE. One announcement I want to make: if you are interested in attending this event, please send me an email. For example, the SynBio seminar series did kick off on August 26th with Francis Arnold, Nobel Prize winner in chemistry, and EBRC and Biomate and Teresa Good, US NSF MCD division director. And also on September 2nd with Jeff Gordon, microbiome father. Speakers in this conference, including Chris Boyd, my advisor, and Patrick Rose, ONR, and Matthew Chang, also the speaker for my seminar. This is basically free for anyone interested in synthetic biology. Also, I'm a council member of EBRC, the sponsor of this conference, and we are organizing climate and sustainability roadmapping workshops that are open to non-EBRC members and very relevant to this audience in this meeting. This is again a free event and you can make your voice heard. Another thing to share, I learned this by my experience, but it took me almost 50 years to realize this by heart. Jeff's quote touched me so much. Unfortunately, I saw this slide late July this year. I wish I saw it earlier. For example, African proverb basically said, to go fast, go alone. But to go far, go together. That's why I'm here to go together with you, all of you, because that basically allow us to improve further, improve the world together. Attention, support, compassion, kind words, empathy, and forgiveness. One last thing, I declare some conflict of interest, and also I'm happy to say Tatenda Chopra from Zimbabwe, of course the African country, is my lab alum, and he basically developed Pfizer COVID vaccine for all of us. And I'm very proud of him. And then you can do it. And he has passion to improve the entire world. And then you can do it together. Thank you so much for your attention. I'm happy to answer any question you may have. Thank you.